We want to be able to simulate a cell like you would simulate a bridge, a road. We have really great tools and an understanding of those kind of physical systems. The biological system is a lot different. It's much more complicated in ways. It's a much more of a black box. And so we really rely on these kind of interdisciplinary perspectives and tools to come together to try to make the rules of biology clear.
My lab is interested in how cells make decisions, and it's really been exciting over the last few years to see our ability to regenerate human blood stem cells. These are stem cells which can reformulate all the blood-forming systems in our body, and more specifically and recently, formation of the human immune system, including T cells and other cells like that. And what this allows us to do is, first of all, modeling human immune system outside the body, which is not currently possible. The second one is using stem cells and stem cell-based tissues to discover new drugs. And finally, there's a new pillar of medicine where we use cells as living therapeutics or cellular therapeutics. And we've been working in that area to use either blood stem cells or T cells as active cells as therapeutics for diseases such as leukemia or other types of cancers.
My research group is conducting high-order engineering of genomes and cells. And one project is a tiny video camera made of DNA, RNA proteins within the cell, such that the cell has a capability of observing what's happening within the cell, outside the cell, or within the body.
In our lab, we're really excited about understanding how microbes that live in our gut affect our health. And through biomedical engineering, what we can do is to try to get back microbes that are disappearing because of industrialization and changed lifestyle that we've had. But we also try to use microbes as a way to predict when diseases are going to occur.
We study how the immune system should work when it's functional and try to help the patients where their immune system might be impaired due to either genetic disorders or disease or aging. So hopefully try to expand the applicability of immunotherapy to broadly benefit more human patients.
The lab's broken into about two domains. One group works on a cancer therapeutic. It's a stem cell marker that we find is upregulated on virtually every cancer when they become metastatic and life-threatening. And we've worked out a way to try to target that protein for therapy. The other side of the lab works on chronic inflammatory disease. We found some new cells that we think regulate that in a variety of different tissues. Now we're working on ways to tweak those cells in ways that will improve things like Crohn's disease or multiple sclerosis. And we found ways to trigger those cells to improve the outcome.
My lab works on RNA vaccines and therapies. The way that RNA vaccines work is by using a patient's cells as a bioreactor instead to make the protein and then train your immune system. The benefit of that is that in the same way that we're able to make really affordable RNA vaccines and scale them and distribute them, we'd be able to do this for therapeutics as well. And so this could potentially make it a lot more accessible for patients.
What my research lab is doing is generating a lot of data by putting in new DNA sequences into human cells in a dish and having those cells annotate that DNA for what it would do in that cell. The more data you have, the better things will get. That's basically our goal in DNA space. We're making more and more data so that we can train better models to understand how our genomes work better.
And the fact that the things we do today aren't going to be decades away from getting into the clinic and helping people, but really could be in the next year or 2 years is just phenomenal to me, and it's something that keeps me excited and wanting to stay involved in the field.